I carry 2 copies of ApoE4, and these 11 insights just changed how I think about prevention, and I'll show you how to use them. Welcome back. I'm Dr. Kevin Tran. I'm an ApoE4 carrier, doctor of pharmacy, and the founder of the Phoenix community. In this episode, I'm breaking down 11 cutting-edge insights that every ApoE4 carrier needs to know directly from the Alzheimer's Association International Conference on ApoE and Lipid Biology that happened in March 2025. So this is very fresh news. These aren't abstract research findings. These are real discoveries about how microglia creates plaque, how ApoE4 accelerates tau spread, and how lipid metabolism might hold the key to early prevention. We'll move fast, one insight at a time, but by the end, you'll have a clear map of what's driving Alzheimer's in E4 carriers and how we might interrupt it. Let's dive in. Before we dive into the science, let's take a moment to understand 2 critical cell types in your brain that play very different roles in Alzheimer's disease, especially if you carry ApoE4. Because to truly grasp the breakthroughs in today's video, you need to understand this. Alzheimer's isn't just about dying neurons, it's about the support systems that either protect or betray those neurons. Let's meet the 2 most important players, astrocytes and microglia. Astrocytes are the brain's support crew. If your brain were a bustling city, astrocytes will be the urban planners and sanitation workers. They make sure everything flows smoothly. Nutrients delivered, toxin cleared, and systems regulated. They are star-shaped, that's why they are named astrocytes, and they wrap around blood vessels and neurons like a safety net. They basically feed neurons, clear the waste, maintain the blood-brain barrier, and produce ApoE in the healthy brain. But under stress, especially in ApoE4 brains, they can flip into a reactive state, pumping out inflammatory signals instead of support. So when astrocytes malfunction, neurons don't just suffer, they basically get abandoned. A second very important cell type is the microglia. Those are the brain's immune defense. Picture them as the special forces unit of the brain's immune system. They are small but vigilant. They're constantly scanning for threats, and they clean up debris, fight off invaders, and they help prune synapses to keep brain networks sharp. In early Alzheimer's, they help clear amyloid and tau, but over time, in ApoE4 carriers specifically, something goes wrong. Instead of calming inflammation, they start fueling it, and instead of cleaning plaques, they may cement them in place. It's like having firefighters who start lighting fires instead. All right, let's go back to the conference and the science now that we have the basics. Dr. David Holtzman opens with a powerful thesis. Yes, many cells in the brain make ApoE, but the real damage seems to happen through microglia.
Different cell types that make ApoE in the central nervous system are probably very relevant for its effects, but many of the likely important pathological effects appear to converge on microglia.
This is a critical shift. Instead of asking who makes ApoE, the field is now asking where does it cause the most damage? And increasingly, the answer points to microglia. In other words, microglia may be the fuse that turns a risky gene into an active disease. So what does this mean for ApoE4 carriers? If you carry ApoE4, your immune cells aren't just passive observers. They are frontline actors, and their behavior could tip the scales between brain resilience and neurodegeneration. That's why at the Phoenix, we don't just look at cognition, we look at the whole system. Inflammation markers, lipid metabolism, and cellular feedback loops that shape how microglia respond. Because if we can shift microglia back into their protective mode, we're not just managing symptoms, we are rewiring the trajectory of brain aging itself. Let's move on to the second insight now. Let's talk about the versions of the ApoE gene, because not all ApoE types are created equal. You've probably already heard of ApoE4 since you probably carry it, the one that raises your Alzheimer's risk, but there are 2 other major versions, E3 and E2, and they don't just carry different risk, they fundamentally reshape how your brain ages. Think of these isoforms like operating systems. E2 is the ultra-secure version. It's rare, but remarkably protective. E3 is the stable default, neutral with no major risk of benefit, and E4 is the high-risk, beta, fast, unpredictable, and prone to critical crashes. Dr. Holtzman lays out the numbers, and they are striking.
If you're E44, about 55% of these individuals already have amyloid deposition. If you have one copy of E4, it's about 20%, no E33 about 15%, and a copy of E2, none of the people that we studied in this population of over 200 people had any amyloid deposition at this age.
That's not theoretical. That's data from actual living brains scanned for amyloid plaque before symptoms even show up. Let's break this down. If you have 2 copies of ApoE4, like I do, you are in a group where more than 1 in 2 already have early amyloid buildup, no matter how young you are, often decades before Alzheimer's is diagnosed. Even 1 copy pushes that number up significantly. And if you have an E2 copy, your risk is so low that in Holtzman's study, none of the participants showed any amyloid at this age. That's like standing at a fork in the road, where your ApoE type determines whether you are heading towards a cliff or a coastline. So if you carry ApoE4, especially E4, there is a real possibility that changes are already happening in your brain, even if you feel sharp today. And that's not about fear, it's about foresight, because Alzheimer's is very slow moving. By the time symptoms appear, pathology may have been building up for 10 to 20 years. That's why we push for early intervention. At the Phoenix, we help members track cognitive biomarkers, lipid profiles, and inflammation before damage sets in, so you are not reacting late, but acting early. The takeaway here is extremely simple. Know your genotype and don't wait for symptoms to act. The earlier you understand your risk, the more power you have to change your trajectory. Cool. Moving on to the third insight. No ApoE equal no amyloid. Here's a wild thought. What if the very gene we've been focused on didn't exist at all? In one of the most eye-opening experiments, researchers asked if ApoE accelerates Alzheimer's, what happened when you remove it entirely? And the result was shocking.
When they simply removed ApoE by crossing these mice to ApoE knockout mice, this blocked the formation of fibrillar amyloid.
In other words, no ApoE, no plaques. Even in a mouse model that was genetically programmed to develop Alzheimer's, that's like removing the fuel and watching the fire die before it ever starts. To understand why this matters, we need to talk about 2 forms of amyloid. You have the soluble beta amyloid. So these are tiny floating fragments that are highly toxic to neurons. Think of them as microscopic splinters, damaging cells from the inside. And then you have the fibrillar amyloid. These are the sticky, hardened plaque that accumulate between neurons over time. They are less toxic per se, but they are a sign the cleanup system is overwhelmed. Now here's the kicker. Removing ApoE didn't just prevent plaques, it also reduced the soluble beta amyloid, especially the dangerous form associated with ApoE4.
Soluble A beta was highest in the presence of E4, lower with E3, and lower with E2.
So the more E4 you have, the more beta amyloid floats freely and wreaks havoc. E3 is better, E2 is best. No ApoE, better still in this specific context. That is very important, right? We're talking about not a clinical setting, but we're talking in a lab setting. This is a dose-response relationship, and it's very telling. It suggests that ApoE4 doesn't just correlate with Alzheimer's, it drives it by actively promoting amyloid production or impairing its clearance. This research is a double-edged sword. On one hand, it confirms that ApoE4 is a key amplifier of amyloid toxicity, which helps explain why our risk is so much higher. But on the other hand, it opens the door to powerful new strategies because we can, 1, reduce ApoE4 expression via antisense oligos or epigenetic tools, for example. We can 2, block its harmful interactions with amyloid, or 3, even mimic the protective traits of ApoE2. We might be able to stop Alzheimer's at the molecular source before symptoms even begin. And that's why we need to focus on real strategies like diet, fasting, exercise, supplements. Those modulate these pathways while we wait for clinical grade therapies to mature. So no, we can't knock out ApoE in humans yet, but what this teaches us is crucial. ApoE4 isn't just a passive risk factor, it's an active engine in Alzheimer's development. And that means we can intervene. The fourth insight is about astrocytes and microglia and how it is a double hit. If ApoE4 is the match, then astrocytes and microglia are the kindling and the oxygen. This next discovery is about pinpointing where, inside the brain's complex ecosystem, ApoE does the most damage, and what happens if you cut it off at the source. Let's start with the astrocytes. So astrocytes are often overlooked, but they are critical. They are the brain infrastructure crew managing fuel delivery, waste disposal, and the blood-brain barrier. And in healthy brains, they are the main source of ApoE. So what happens if you only remove ApoE from astrocytes while leaving every other cell untouched?
When you remove astrocyte-only E3 or astrocyte-only E4, this strongly blocked A-beta deposition and fibrillar A-beta deposition to a very similar extent to what you see in an ApoE knockout.
That's huge. Even without touching other cells, just silencing ApoE4 in astrocytes was enough to dramatically reduce amyloid plaque. In other words, astrocyte-derived ApoE might be the spark that ignites Alzheimer's in the first place. Then, the scientists looked at microglia, the brain's immune warrior. And here's where things got even more surprising. If you remove microglia before amyloid even appears, plaque formation gets shut down, just like with ApoE knockout.
And if they remove microglia before amyloid forms, it really blocks most of the amyloid deposition in the brain that's going to occur, at least in the parenchyma. So one question is, this is a very similar to effect that you see if you knock out ApoE completely.
So let's piece all of that together. Astrocytes may start the fire by releasing ApoE that encourages amyloid buildup, and microglia may fan the flames by responding in overly aggressive inflammatory ways. And if you interrupt either one, the fire never takes hold. This is a double hit model of Alzheimer's initiation. Astrocytes release ApoE, which binds the amyloid beta and promotes its aggregation. Microglia react to this buildup, but in ApoE4 brains, their reaction is dysregulated. Instead of resolving the damage, they fuel it. These two cell types don't act alone, they collaborate in the disease. And if you silence ApoE in just one of them, you dramatically reduce pathology. So what can we do about this? It gives us two powerful ideas. One is precision targeting. Future drugs might not need to erase ApoE entirely, just lower it in the right cell types at the right time. The second idea would be lifestyle leverage. Exercising, fasting, omega-3s, anti-inflammatory habits already affect astrocyte and microglial function. We can start modulating this system right now. Moving on to insight number 5 about microglia and how they create the plaques. Now here where things get wild, and honestly it's a bit unsettling. We've long assumed that amyloid plaques simply accumulate in the space between neurons like sticky trash piling up on the street. But Holtzman's team flipped that theory. They found that plaques don't start in the extracellular space, they form inside the microglia first. That's like discovering the garbage isn't being dumped on the streets, it's being manufactured inside the cleanup crew's headquarters. I love these analogies.
A-beta aggregation occurs within microglia, at least in these mouse systems. It forms fibrils there, and ApoE also gets in the microglia. It forms fibrils itself, and it's required for this A-beta fibril formation.
Let's unpack that. Microglia don't just react to amyloid, they are the site where it forms. ApoE gets pulled into the microglia, and once it's inside, it helps the beta amyloid clump into the toxic fibrils that eventually become plaques. So instead of being innocent bystander, microglia are functioning like factories, actively building the very thing they're supposed to destroy. Now you might ask, what happens if you try to build a plaque without either of those components? Well, that's exactly what the scientists tested.
And they found that if they added ApoE back into the brain, it seeded A-beta again, looked like in microglia. But then if they took the amyloid depositing mice in the absence of ApoE, but they removed microglia, they could not seed A-beta anymore.
Let that sink in. Without ApoE, no plaque formation. Without microglia, same thing, no plaques. Even if you reintroduce ApoE, it's useless without microglia present. Both are essential partners in crime, and this is a key paradigm shift. So we used to think of ApoE and microglia as separate players in this complex system, but this research shows they are co-conspirator. ApoE enters the microglia. Inside it, it enables amyloid fibrils to assemble. These fibrils then exit and accumulate into full-blown plaques. This doesn't just reframe Alzheimer's as a disease of accumulation, it reframes it as a disease of dysregulated internal processing. What it means for you? Well, you just need to track early inflammatory signals, especially in the brain. So basically managing your lipid metabolism, exploring interventions that might calm microglia or reduce ApoE4 expression. Because the earlier we understand these internal dynamics, the more precisely we can then intervene. The takeaway is sobering but powerful. Alzheimer's plaque don't just happen to you, they may be happening inside you, driven by two key players, ApoE and microglia. Let's move on to insight number 6, the Christchurch mutation and natural shield. Sometimes science doesn't move forward with a new drug or a new mouse model. Sometimes it's one person who rewrites the playbook. This is the story of a woman in her 70s, part of a Colombian family with one of the world's most aggressive Alzheimer's mutation. She should have developed dementia in her 40s, but decades later, she was still sharp, independent, and fully functional. And when researchers scanned her brain, what they saw left them stunned.
She had a lot of amyloid deposition as seen on amyloid PET scan, but when a tau PET scan was done, you can see that all these areas that normally would develop tau pathology at this stage of disease were not it wasn't there.
This is unheard of. She had massive amyloid plaques buildup, the kind that typically guarantees a rapid decline, but the second protein in Alzheimer's, the one most tightly linked to symptoms, tau, was nowhere to be found in the region it normally invades. It's as if the fuse was lit, but then the fire never spread. So researchers traced this resistance to a rare genetic variant in ApoE gene, E3 Christchurch. And what they found is astounding.
In the presence of the Christchurch mutation, this seeding and spreading around individual plaques was suppressed by about 70%.
This wasn't a fluke. This variant actively blocked tau from propagating across the brain, even in an environment full of amyloid. And that's important because it supports a powerful emerging model. Amyloid triggers Alzheimer's, but tau drives the symptoms. Stop tau from spreading and you stop the decline. But the mystery didn't end there. Researchers wanted to know how the Christchurch variant pulled this off. What was it doing differently at the cellular level?
Further, if you then waited till the myeloid cells filled up with tau aggregates and then followed the aggregates over time, you could see that the Christchurch myeloid cells degraded tau faster.
So the real magic wasn't just the gene, it was the behavior of microglia. Christchurch microglia weren't passive or toxic, they were elite cleaners. Faster tau uptake, more efficient degradation, and less release of seed-competent tau, which means fewer copies to infect neighboring neurons. That's like having super janitors who not only scrub the floors, but they clean so fast and so thoroughly that contamination never spreads. So this case changes everything because it shows that even with massive amyloid burden, Alzheimer's isn't inevitable. If you can stop tau from spreading, you stop the damage. And more importantly, it proves that microglia can be reprogrammed from destructive agents into guardians of brain health. For those of us with ApoE4, that raises a crucial question. Can we mimic this Christchurch effect? Maybe not genetically, but through lifestyle strategies that reduce inflammation, through supplements that boost lysosomal function, and soon through drugs and gene therapies that tilt microglia into a protective state. This is giving us a lot of hope, and then we're moving on to insight number 7, ApoE versus tau. That's like a molecular traffic jam. So if we zoom in, like way into the molecular level, because sometimes what determines your brain's fate comes down to who gets through the door first. Inside your brain cells, there's a receptor system, LRP1 HSPG, that's a mouthful, that acts like a VIP entrance for cellular cargo. Both ApoE and tau protein use this same doorway to get into cells, where they're supposed to be processed and then cleared. But here's the catch. ApoE competes with tau for the access, and when there's too much ApoE, especially ApoE4, it acts like a traffic jam at the entrance. The cell's cleanup crew cannot get to tau because the line is already blocked by ApoE.
Previous studies had found that tau aggregates are a ligand for LRP1 HSPG pathway. The Christchurch variant doesn't bind very well to the HSPG LRP1 receptor system. So what Yin shows here is that if you add ApoE3 particles, this decreases the uptake.
This is such a subtle, elegant mechanism, and it explains so much. The Christchurch variant changes ApoE structure just enough that it no longer hogs the LRP1 HSPG doorway, so the VIP door. Instead of jamming up the system, it politely steps aside, allowing tau to enter the cell and get degraded. Think of it like this. ApoE4 is that person who double parks and blocks the entire street, while ApoE3 Christchurch is someone who parks cleanly and lets the ambulance through. This tiny tweak allows the system to work as intended. tau gets taken up, degraded, gone. No lingering seeds, no spreading, no cascade into Alzheimer's pathology. So what does it mean for us ApoE4 carriers? Well, it changes how we think about treatment. For years, researchers have been trying to reduce ApoE4 levels entirely with antisense therapies, gene silencing, knockouts. But this insight suggests a more refined approach is possible. Maybe we don't need to get rid of ApoE. Maybe we just need to change how it behaves, specifically how it interacts with tau clearance pathways. In other words, instead of tearing down the road, we reprogram the traffic pattern. Cool. If you are learning something new right now, hit subscribe. These breakdowns turn complex science into tools you can actually use as an ApoE4 carrier, so don't miss any of my videos and subscribe to this channel. Moving to insight number 8, about the double hit microglia plus ApoE fuel tau damage. So far, we've seen how ApoE and microglia play key roles in amyloid buildup. But what about tau, the protein that actually drives neuron death and cognitive decline? Well, Holtzman's team explored this by using tau-pathy models, special mouse model designed to mimic the second half of the Alzheimer's equation. And what they found is crystal clear.
In the presence of any form of ApoE, E4, E3, or E2, you get a lot of damage in parts of the brain that develop tau pathology, including hippocampus and entorhinal cortex. But what was really striking is that when these mice were crossed to ApoE knockout mice, there was virtually no injury that occurred in the brain.
So even in E2, the protective version of ApoE worsens tau-related damage in this model, removing ApoE altogether, and the brain becomes dramatically more resistant to damage. So what's happening here? This shows that ApoE, regardless of isoform, acts like an accelerant in tau-driven disease, not necessarily by causing tau, but by making the brain more vulnerable to its effect. It's like the same spark hitting different materials. With ApoE present, the fire spreads. Without it, the spark fizzles out. But Holtzman's team didn't stop there. They asked, what happens if we also remove microglia, the very cells we know are involved in tau uptake, degradation, and inflammation?
And when you remove microglia with a CSF1 receptor inhibitor, that blocks the injury. And if you remove ApoE, it's neuroprotective. If you do both, it's very neuroprotective.
So this is huge. I feel like this is a topic we covered across this entire conference. So the researchers actually found these a bit independently. When you remove either ApoE or microglia, you reduce brain damage. But removing both, you unlock maximum protection. The combined effect is stronger than either one alone. It confirms a synergistic toxicity model. ApoE makes neurons more sensitive to tau. Microglia amplify that sensitivity with inflammation and faulty processing. Together, they create a feedback loop that accelerates neurodegeneration. So as an ApoE4 carrier, you want to have interventions that reduce both arms of this toxic synergy through fasting, immune modulating routines, DHA supplementation, low inflammation diets, and potentially soon ApoE silencing therapies. So let's put it plainly. If you carry ApoE4, your brain may be fighting on two fronts, tau toxicity and immune overreaction. But this isn't a cause for fear, it's a clear signal that your strategy needs to be multi-layered, targeting both inflammation and gene expression. Insight number 9, let's talk about something that gets a bad reputation: fat. In most contexts, we think of fat as something to avoid, but in the brain, especially inside microglia, fat stored as lipid droplets can actually be protective. These lipid droplets are like biological shock absorbers, little storage bubbles that tuck away excess cholesterol, fatty acids, and cellular waste, keeping the rest of the system stable. But what happens when microglia loses that ability to store fat properly?
Removing the ability of microglia to form lipid droplets caused more damage. The hippocampus is smaller, ventricles are larger, more p-tau staining in the brain.
Well, that's a big deal. When microglia can't form lipid droplets, the brain visibly degenerates. The hippocampus, that's the memory center of the brain, shrinks. The ventricles, those are fluid-filled gaps, swell up. And lipid waste piles up in all the wrong places. So this isn't just poor housekeeping, it's structural damage. But what's even more interesting is where all that fat ends up.
When you remove the lipid droplet formation, you actually see an increase in lipid accumulation in microglia, again, overlapping with staining for the microglial endosomal lysosomal system, not within PLIN2 positive vesicles.
Instead of being stored safely in droplets, lipids are now shunted into the endosomal lysosomal system as the cell's internal trash compactor. But here's the problem. That system wasn't designed to store fat. It's like trying to stuff oil into your paper shredder. This overload cause microglia to become dysfunctional and stressed microglia, as you know by now, are exactly what fuels neurodegeneration in Alzheimer's. So this insight connects two major systems, lipid metabolism and immune function. Microglia need to handle fat properly to stay calm and do their job. When they lose their ability due to genetics, aging, or inflammation, they start to malfunction, becoming overly reactive, inflamed, and unable to clear tau or amyloid properly. And in ApoE4 brains, well, lipid handling is already compromised, so this lipid droplet breakdown creates a perfect storm. Microglia are both exhausted and become toxic. So what does it mean for you, ApoE4s? Well, supporting lipid droplets formation may be a therapeutic strategy. DHA and omega-3s known to integrate into brain lipids could help buffer these microglial stress. Fasting, ketones, and mitochondrial support will help improve lipid handling and energy use, and targeting the lysosomal system could become a future direction in drug development. Cool. I hope you are following this. Insight number 10, it's about lipid export, the microglial escape hatch. We've seen what happens when microglia can't store fat properly. It overwhelms their internal system, triggers stress, and accelerates brain damage. But here is the hopeful part. Some microglia don't collapse under pressure. They manage to adapt. Even when lipid droplet formation is blocked, some microglia find another way. They activate a backup system, a cellular escape hatch, to export lipids out of the cell and restore balance.
When we remove lipid droplet formation in microglia, you see an increase in cholesterol esters and BMPs and other lipids within the brain of these animals. And the microglial disease-associated microglial phenotype is present in the tau mice, but in the absence of lipid droplet formation, they have an increase in the subpopulation of microglia that overproduce ABCA1, ABCG1 lipid export machinery, probably trying to compensate for this added lipid burden in these cells.
So let's break that down. CD68 is a marker of lysosomal activation, meaning the trash compactor system is working overtime. Cholesterol esters are building up, indicating the cell is overloaded. So what does it do? It flips on its lipid export pathways, trying to push the excess cholesterol out to survive. And this is the cellular equivalent of hitting the emergency vent button before the whole system overheats. So this adaptability is crucial. It shows that microglia aren't just passive victims of Alzheimer's, they have tools to recover. But the success of those tools, of that response, likely depends on your genetic background, your lipid profile, and metabolic flexibility. And here's where ApoE4 comes in, because ApoE4 impairs lipid transport. It binds lipid less efficiently than E2 or E3. Then it reduces the function of receptors, which are essential for cholesterol export. So even if your microglia wants to adapt, they may be less able to do so simply because ApoE4 gets in the way. That's why strategy to enhance lipid exports are so promising, especially for us ApoE4 carriers. So what does it mean for us? It gives us a clear message. The key isn't just avoiding stress, it's also to build resilience inside the system, and lipid export is a perfect example of that. So you have to look at your HDL function, basically how well your body clears fat, your omega-3 to omega-6 ratio to reduce inflammation and support membrane health, and even potentially experimental markers tied to lipid export and reverse cholesterol transport. Because if you carry ApoE4, you can't just assume your lipid system is working optimally like E3 and E2s. You have to support it deliberately. Cool. Moving on to the last insight, the master blueprint, ApoE, microglia, and the decline. After these 10 findings, we close with one final insight. And it's not just a summary, it's a diagnosis of the system itself. Because whether you're looking at amyloid plaques forming, tau spreading between neurons, or lipid systems breaking down, the common thread is always the same, ApoE and microglia. We've been covering this all along for this entire video.
I think it's clear ApoE is the most important genetic risk factor for late onset AD.
Let's pause here. This isn't just about having a risk gene, it's about carrying a blueprint that rewires your brain's most fundamental system, from immune function to metabolism to cellular cleanup. And if you carry ApoE4, that blueprint is more aggressive, more inflammatory, and more disruptive, especially inside microglia. So this insight ties everything together. ApoE4 doesn't act alone. It reshapes how microglia behave. Microglia aren't just immune cells, they are central processing hubs for amyloid, tau, and lipid balance. And when that system goes off course, the damage accumulates long before symptoms appear. But here's the silver lining. If we can map this pattern, we can then interrupt it. This is the essence of precision prevention. It's not about waiting for the disease. It's about intervening very early in the right pathways, in the right cell. All right, we covered a lot of insights today. Let me give you a quick recap now. These 11 core insights on how to reshape how we understand Alzheimer's in ApoE4 carriers. Insight 1 was about that all roads lead to microglia. No matter which cell makes ApoE, the damage converges on microglia, your brain's immune cells, which can either protect or accelerate degeneration. Insight 2 was about ApoE isoforms, the risk by type. E4 drastically increases your early amyloid buildup, often decades before symptoms, while E2 appears protective. So genotype, of course, matters, but normally, if you're here, you should already know that. Insight number 3, no ApoE means no amyloid. Knocking out ApoE in mice stops plaque formation completely, proving ApoE isn't just associated with Alzheimer's, it drives it. Insight number 4 was about astrocytes and microglia that work together as a double hit. ApoE from astrocytes starts the fire, microglia fan the flames, and disrupting either one, and you slow down plaque formation. Insight 5 was about microglia creating those plaques, because they don't form passively, they start inside the microglia. ApoE and microglia are co-creators of early Alzheimer's pathology. Insight number 6 was about the Christchurch mutation, a natural shield. Remember that Colombian lady that defied Alzheimer's despite heavy amyloid because her microglia cleared tau more efficiently thanks to this rare protective ApoE variant. ApoE versus tau, that was insight number 7, that molecular traffic jam, if you remember the VIP door. Basically, ApoE competes with tau for cellular clearance. The Christchurch variant steps aside, letting tau be cleared before it spreads, while when you don't have it, ApoE blocks the road, blocks the door, and then tau can't be cleared. Insight number 8 was about, again, another double hit where ApoE and microglia fuel the tau damage that is later in the pathology. Basically, removing either ApoE or microglia and tau-related damage drops. If you remove both, you get maximum protection. Insight number 9 was about those lipid droplets that are not just fat, but safety nets. So microglia need lipid droplets to stay stable. Without them, the brain degenerates, especially in E4 carriers, especially reverse ApoE4 carriers where we have very poor lipid handling. Insight number 10 was about lipid export. So that microglial escape hatch. So even stressed microglia, they try to adapt by exporting the fat outside, but ApoE4 impairs this system, making support for lipid metabolism critical. And insight number 11 was the master blueprint, how ApoE4 actually rewires microglia to drive damage across all fronts. We talked about amyloid, tau, lipid dysfunction, but those patterns can be interrupted. Cool, I hope you like those insights. They show us how damage happens, where to intervene, and why waiting for symptoms is way too late. This is why early action matters, why prevention must be extremely precise, and that's why inside the Phoenix community, we help members track the right biomarkers, design structured self-experiments, and build daily systems that mimic the most protective biology we've seen, from fasting to DHA to lipid modulation. We've covered DHA's role in myelination in another video. We'll keep breaking down the science in future deep dives. But for now, here's the most important thing. If you are an ApoE4 carrier and want to beat the odds and defeat Alzheimer's, check out the Phoenix community. The link to apply is in the description below this video. I review every application personally to make sure that you're a great fit. So if you're coming from YouTube, just mention that in your application form and I'll make sure to show you some little extra love. And thank you for watching. More importantly, thank you for taking your health into your own hands. I'll see you in the next video. Bye.